Thanks, Samir, for that excellent presentation. We now have Dan Theodorescu. He's there. Okay, great. Dan is the director of the Samuel Oschin Comprehensive Cancer Center Institute at Cedars-Sinai in LA. And Dan will give yet another academic perspective on selecting preclinical combinations.
Great. Well, good morning, and thank you for inviting me to give this presentation. It's really a wonderful meeting. We learn a lot. My interest has been bladder cancer for the past 20 years. And so we've taken a number of approaches to really identify the drivers of bladder cancer, specifically the drivers that actually lead to metastatic disease and tumor progression. So I think this slide speaks for itself. Bladder cancer is one of those diseases that presents in two fairly distinct forms with different molecular characteristics. And basically, one form, which is the most common form, actually is if you will, a recurrence-based disease that doesn't often kill patients with a very good 5-year survival. Unfortunately, the other form is a lot more aggressive, but fortunately, it's only 30% of the population that get bladder cancer and leads to a 50% 5-year survival, with the majority of the patients that do succumb from the disease succumbing from metastatic disease. So chemotherapy has been used for many, many years. And you can see here the outcome of combination chemotherapy with a GEM-CIS or MVAC, 4-drug combination, has been, there are some survivors, but not many. Checkpoint inhibitors have been used in bladder cancer, one of the first tumors to show dramatically improved results. But as you can see here, from both of these, there's still room for improvement. Of course, the previous talks that you've heard really set up the rationale, and I think nobody will argue the point that there is still room for improvement in this field. So the question is, how do you improve this? Well, you can discover completely new therapies or build upon the therapies that we have at hand, both chemotherapy and immunotherapy. So there are multiple ways to do this, of course, to build upon these therapies. And you've heard and you will hear for the rest of the next 2 days various approaches to do this using patient material, experimental systems, et cetera. And you can also use logical combinations from the literature and try to combine them. And of course, those may or may not work. So we thought about this a fair amount and decided really to let the cancer speak to us. And without any biases or preconceived notions, use technologies that would actually query the biology and get an answer from the cancer, if you will. Oh, that rhymes, doesn't it? An answer from the cancer, maybe I'll use it again, to tell us what therapeutics to use. So here's really the cartoon that underlies the concept. And the idea is to use murine models, some of which we developed in our own laboratory, that recapitulate and have similar genetics to human cancers, and then use technologies to use unbiased determination of the pathways that maintain a resistance to checkpoint inhibitors. And then ideally, some of those pathways can have small molecules that could be used to in combination with checkpoint inhibitors. Obviously, these are not the only things going on here, but this is more of a schema to get the framework to understand what I'm about to show you next. So the idea then is to use technologies that take checkpoint inhibitors and then at the same time cause blockade in these various different signaling pathways. And for example, here, it shows the idea that you use checkpoint blockade, the tumor itself doesn't do anything. If you block only one of the pathways that leads to a certain resistance phenotype, nothing happens. But if you do both at the same time, the tumor, the cancer cell dies or gets destroyed, et cetera, but it disappears or gets reduced in number in the actual tumor. So what we decided to do is apply a technique that we had successfully used in defining some tumor suppressors and metastasis suppressors in the past. And so here I'm going to describe to you in this slide what we did. So again, focused on bladder cancer, we wanted to show proof of principle, but you can conceptualize the idea that this could be applied to other cancers, other scenarios. So because we wanted to focus on bladder and we had the models in bladder, we went to the clinicaltrials.gov database, and as you can see here, we searched for all the trials in bladder cancer, and then we went through a number of parsing by hand steps to really identify a subset, a tractable subset of drugs and that were used in bladder cancer. And there are multiple considerations here, and I'm happy to answer questions if there are afterwards. But the idea was we wanted to get something, we wanted to do a proof of principle. So it wasn't as broad as we could now do given the results I'm going to show you. So we parsed them fairly aggressively to come to a smaller number. Of course, one of the critical considerations was that they have, these drugs have targets, and these targets are also found in mouse tumors. So we ended up with 192 genes and then constructed an shRNA barcoded library and infected murine bladder cancer cells. And this is a cartoon, a theoretical cartoon of some of the possibilities. We have one lentivirus per construct, multiple redundant constructs. Each cell takes up, on average, one lentiviral particle. So one gene gets knocked out in each cell. And then this is a pool population. So you reconstitute the transfected population, and you then inoculate mice with the cells that are basically containing the repertoire of knocked or reduced gene expression based on the shRNA. Okay, so then you do this injection in the mice, and then you take the mice and then you randomize them to treatment. And we've done multiple checkpoint inhibitors, but I'm going to show you, since the topic is PD-1, PD-L1, I'm going to show you what happened with PD-1. And so we treated mice with PD-1, and then let the tumors go and remove the tumors at various time points. And then this is a cartoon again. The idea is that I want to emphasize is that the tumors basically have a different composition of cells that have different shRNAs in them. So the distribution of the shRNAs in a controlled treated tumor versus one that has been treated with PD-1 is not the same. And how do we know this? Well, because after taking the tumors out, we basically prep them and then use next generation sequencing to identify, if you will, the composition or the shRNA count of control versus the treated tumors, and then you compare. And the idea is that shRNA constructs that are no longer found in the treated tumor indicate genes that when their expression is reduced, they're at, for whatever reason, ideally because they're being destroyed by the immune system, are in lesser quantity in the treated tumor. So if we do that analysis, we ended up with 32 genes, which is actually a little more than I was thinking, but I was happy with that result. And I'm going to show you one of these genes today in this brief talk. And this gene is DDR2, which parenthetically is a gene that's is mutated in lung cancer. So what is DDR2? Well, here it is. It's a collagen receptor, and it's not a very thoroughly studied gene. So the exact mechanism of action, how it regulates cell proliferation, adhesion, migration, metastasis in various models and various tissues, exactly downstream is not that clear, but now we can add potential immune modulation to this. The other reason why I'm showing you this is because there are pharmaceutical agents that do reduce activity of this kinase, although they are not, at least the ones in clinical use, which is the dasatinib, are not specific to DDR2. So what we did next is look at DDR2 in bladder cancer, and this is basically an expression profile that we got from the literature in multiple different data sets. And the bottom line is, as you can see here, DDR2 is amplified, and actually this is from the CBio portal. Bladder cancer shows significant amplification. This is among all cancers. Then we looked at expression of DDR2 in various studies in bladder cancer, in muscle invasive versus non-muscle invasive. So the two types of bladder cancer, the more aggressive and the less aggressive tumors. And then we looked at DDR2 expression as a function of outcome, again in various studies, and DDR2 high expression is a bad prognostic factor. So tumors that have it do worse, at least bladder tumors. So then we wanted to look at exactly DDR2, what it does specifically in the context of checkpoint inhibitors and specifically PD-1. So we looked at generating a variety of cell lines. We obviously looked at bladder first, the line that we identified DDR2 here, and we transfected an shRNA, and then we also did CRISPR with similar results. And we have the result is right here. When we knock down DDR2 and in the NA13 murine bladder line, you could see the various single drug treatments here, and you could see that the PD-1 the treatment has begun, when the tumors were established, not preemptively. So this is the combination therapy here. We also extended these observations to other tumors, breast cancer E0771, and the combination is here. And interestingly, melanoma B16, which has been a relatively refractory tumor, is shown here with the combination showing significant reduction in tumor burden. So then we wanted to study the mechanisms of this, and we're still working quite actively in this space to really identify what is going on. But early analysis of RNA-seq and GSEA has revealed that the tumors that are treated with the combination show an increased activity in T cell and across the immune spectrum, signifying strongly that there's an enhanced response. Obviously, there are caveats with this analysis, because in order to be able to do this analysis, it means you need a tumor. So the animals that had complete tumor disappearance, you recall some of the curves showed a complete cure of some of the combination groups. So that needs to be kept in mind. Obviously, it's not as straightforward to figure out what's going on. So, but in essence, what we're doing in order to overcome that issue is we're doing temporal analysis. We also did CyTOF to really get a sense of what is going on as well, again, with the same caveats at the RNA-seq, and we see that there is a CD8 and cluster 15 enhancement in the combination. So how about bringing this closer to the therapeutics? We then used the dasatinib, and again, obviously with the caveat that the dasatinib is not a completely targeted agent to DDR2, but the shRNA basically addressed the issue of specificity. And you can see here, that in NA13, again, the plots, and B16, again, similar to shRNA. And we did MC38, and the combination is here in orange. So in conclusion then, from this is that I think functional genomic approaches where the tumor speaks for itself without biases to tell you what would be the most effective treatments seem to yield some very interesting and tantalizing results. We're really moving forward to understand the mechanisms underlying the DDR2 effect, but also are pretty excited about the other 31 hits, many of which have small molecule inhibitors. And we also want to extend the results beyond the tumors that we have into tumors that have traditionally not responded to immunotherapy, such as pancreas and prostate, and then strongly considering clinical trials with checkpoint and DDR2 inhibitors in patients. So this last slide really asks the questions that rose, at least in our minds, during this work is, what is the role of preclinical models? Obviously, I'd lie to you if I wasn't excited about the animal results, but one of my mentors when I was in grad school, although I didn't train with him, was Judah Folkman, and I've known him since, the mid-90s, and I remember him telling me then, we've been curing mice of cancer for 20 years, and that was in 1995. So when I see these, I remember those words, and I wonder, then what can we do to make sure that we optimize these preclinical models so their hit rate in actually patients is higher than it is. So those are some of the questions I have, and perhaps we can discuss in the panel. The other more granular question is really what technologies we should use in those models. Should we use humanized mice? Should we use other suppressive techniques in terms of gene functional genomics? And how can we then use that knowledge to actually validate them preemptively before even a trial is done with biomarkers? So I think there are a lot of real questions to be able to, that we need to address in order to be able to optimize these preclinical models. But I suspect that at least some of the data that we're going to get through preclinical models is going to hone in and reduce the complexity or the options for combinations. Thank you.